before forests or animals. Earth turned through space as a dark ocean world, scarred by heat and impacts. From above, the coast might resemble a rugged Pacific shore, but no living thing waited there below in silence. Waves struck black rock. Steam rose from fractures, and minerals moved through water, without any biology shaping them yet. What happens when hot water keeps touching fresh rock? Again and again, for millions of silent years on this planet. Lightning split the storm clouds, adding violent energy to a world already burning from deep below, everywhere at once. At this stage, the story is not about creatures, but chemistry searching through countless possible arrangements across time. The first life was probably not a tiny animal appearing suddenly, fully formed, in a tide pool at dawn. Modern scientists work backward from rocks, chemistry, and cells, carefully reconstructing an event no one alive witnessed directly. Somewhere in that lost world, molecules may have begun acting less like clutter and more like a system. To follow that possibility, we have to leave familiar Earth and enter the planet before biology had begun. The air smelled of minerals and rain, while dark seas rolled beneath skies no bird would ever cross. Every wet stone held surfaces where atoms could stick, separate, and meet again in changing water at the surface. For an American viewer, imagine a national park without trees, animals, soil, or even oxygen, rich air anywhere nearby. Chemistry was not alive, but under the right pressure, it could begin forming temporary order from chaos for moments. The mystery is not where a creature appeared, but when non-living matter began behaving differently together over time. That ancient question still guides laboratory missions and the search for life beyond Earth today, right now. Because no camera recorded the beginning, scientists read clues left in rocks, cells, and chemical reactions with patience. Can an experiment recreate one step toward life without pretending it has solved the whole story by itself? Mars, Europa, and Enceladus matter because they remind us that water alone is not enough anywhere for life. Earth's beginning became part of a larger question. Which worlds can turn chemistry into biology? Eventually, over time, a barren landscape on Mars can make early Earth feel less like history and more like evidence today. Stone remembers conditions that living things cannot, preserving chemistry, pressure, heat, and water in silent layers over ages. Far from sunlight, ocean worlds suggest that rock and water may create chemistry beneath frozen shells in darkness. The most promising places can also be unreachable, buried under ice, pressure, radiation, or miles of darkness below. Habitability is not one magic ingredient, but an overlap of water, energy, chemistry, and stability through time on worlds. Explorers often work like detectives, using instruments to infer worlds their eyes can never directly enter or touch. The deeper problem is not finding water, but explaining how non-life starts building useful patterns in the first place. Earth remains the only place where we know that chemistry actually crossed into living continuity once, for certain. Before cells could evolve, something had to gather molecules, protect reactions, and keep changes from disappearing over time. So the investigation returns to Earth itself, before fossils, before DNA, and before any recognizable organism existed anywhere. Researchers cannot watch the first life begin, but they can search for shadows it may have left directly. Ancient rocks sometimes preserve textures, carbon signals, or layered structures that still resemble biological fingerprints after deep burial. A thin rock layer can hold more deep time than a city block holds human memory in stone. Scientists compare shape, chemistry, and setting because no single clue can carry the whole claim alone by itself. If a pattern looks ancient and organized, is it a fossil or a trick made by geology alone? That caution matters, because minerals can imitate life like forms without any organism ever being present there at all. A possible signal means little without the surrounding rock, the ancient environment, and the nearby chemistry around it. Each test narrows the possibilities, linking a sample to heat, water, minerals, or possible biological activity long ago. The farther we go toward the beginning, the more the evidence becomes fragmentary and contested again over time. That uncertainty does not end the story. It makes the world before biology even more important to understand. Models and experiments cannot replay early Earth, but they can test which pathways remain physically plausible when tested. The investigation needs four broad things: water, energy, chemistry, and enough time for selection, like processes to emerge there. The answer may not be one birthplace, but many environments pushing chemistry in different directions together over time. To understand that possibility, we must step back into a planet with no biology at all once again. Now the investigation leaves modern instruments behind and returns to storms, rocks, oceans, and ancient heat deep below. Before biology, early Earth was still a world of moving water, broken crust, and restless energy rising everywhere. What could happen when rain, tides, and hot ground kept forcing water through reactive stone again for ages? The same harshness that made Earth dangerous may have supplied the energy chemistry needed to change over time. The story now shrinks from oceans and storms to invisible reactions happening on mineral surfaces one layer deeper. The next clue is whether restless chemistry can stop scattering and begin organizing itself into something persistent over time. Wet basalt, rain, and steam cover the shore, but this lifeless ground is already chemically exposed and restless. On the mineral crust, tiny cracks and beads of water create small spaces where chemistry can gather and change. Volcanoes fed early Earth with heat and fresh rock, while making every shoreline unstable, dangerous, and temporary. This small coast belongs to a larger engine, where oceans, atmosphere, and heat constantly trade energy across the planet. Rain runs over volcanic stone, carrying dissolved minerals into low basins and storm dark coastal pools below the cliffs. As the water mixes, chemicals spread through channels, showing motion without suggesting life has appeared here. Yet, like a lifeless Yellowstone pool, this basin is familiar in shape, but stripped of every living trace. Thin films cling to the rock, gathering dissolved material before releasing it again with the next flow. The pools do not stand alone. Narrow channels connect them into a restless prebiotic network of water and stone. The question is whether violence, rain, and heat can still build order when nothing is alive. Above the shoreline, the young atmosphere churns with gases, storms, and chemistry still far from breathable. Lightning tears through the clouds, adding raw energy to the scene, but not magically creating life. Rain pulls dust from the air, moving material between sky, stone, and water with every storm. One drop belongs to a larger weather system, like radar watching storms sweep across an entire continent. Beneath the pool's surface, bubbles rise through minerals, marking a boundary where gases and water meet. A faint overlay shows molecules drifting through runoff, mixing, separating, and disappearing into darker channels below. The same storms that tear surfaces apart may also refresh them with new chemical combinations again and again. Following the water, the viewer traces a clue across an empty world where chemistry keeps moving. Heat rises from mineral ground as shallow water spreads across it, turning the edge into a laboratory. Now the mystery changes if water carries chemistry. What remains after the water disappears into air? In the drying basin, pale rings mark where repeated floods left minerals behind in quiet layers. Evaporation pulls water away, concentrating dissolved material into thin films, where surfaces begin to matter more. The visualization shows concentration, not a finished answer, because no single pathway is proven by science. The basin resembles a desert dry lake bed, where wet and dry cycles can repeat for ages. The camera enters tiny channels where trapped liquid and dissolved chemicals can linger after larger flows vanish. A fresh drop restarts the cycle, dissolving what dryness had just gathered on the mineral surface. Drying is not simply water leaving. It can repeatedly bring scattered materials closer together over time. No ecosystem exists yet, but the terrain behaves like one connected chemical landscape before biology has begun. The open pools narrow into protected cracks, where the next clues are hidden inside ancient stone. If concentration matters, could minerals also shelter reactions long enough for real complexity to grow here? Across the planet, the surface is struck, shaken, melted, flooded, and reset again and again without warning. Impacts could destroy local settings, yet they also brought heat, metals, and fresh reactive material from space. A dark fragment in the mud becomes a clue, not proof that life arrived from space. The overlay suggests possible organic chemistry, while the fragment remains only rock, dust, and non-living material. A hostile surface may seem hopeless, yet each disruption can open new chemical possibilities afterward on Earth. Erosion carries impact dust, volcanic ash, and minerals downhill until shallow coastal water finally collects them. Hot pools, wet shores, ash fields, and basins coexist, giving early Earth many chemical settings at once. The coastline feels almost familiar, but there are no plants, shells, birds, or footprints anywhere at all. The violent landscape narrows again to grains, films, and wet surfaces where chemistry actually touches matter. After so much destruction and renewal, the next question is whether any stable pattern can survive. Early Earth was not one scene, but many prebiotic environments connected by water, heat, and rock. Mineral rings and runoff preserve hints of earlier cycles, like marks left by a repeating tide. Wetting, drying, heating, mixing, and erosion repeat together, turning landscapes into long, running chemical experiments without life. The world is lifeless, but not empty. It constantly rearranges matter, energy, and possibility across its surface. From grains to coastline, the whole region works like a planetary chemical engine, without living parts. From a modern observing view, scientists read these settings as clues, not eyewitness records of the beginning. These environments are possible stages for prebiotic chemistry, not confirmed birthplaces of the first life on Earth. With the settings established, the investigation turns toward the ingredients that chemistry still needed before biology. Early Earth had water, heat, and surfaces, but useful ingredients still had to appear somewhere first. Suspended particles drift in the pool, leaving one question: where did life's raw materials come from? Did life's raw materials form on Earth, arrive from space, or gather from both sources at once? Early Earth was not isolated. It moved through a young solar system filled with restless debris. A meteor crossing the sky carried heat, minerals, and possibly carbon chemistry into the ancient atmosphere below. Every impact could erase fragile compounds, yet also create fresh conditions for new reactions to begin. Destruction sometimes exposed clean mineral surfaces, giving chemistry new places to stick, gather, and slowly change. Organic molecules sound alive, but here they mean carbon-based chemistry, not organisms, microbes, or hidden cells. Think of scattered parts on a kitchen table. Useful pieces are present, but nothing works yet. On tiny mineral grains, faint chemical traces could survive, where open water might quickly dilute them. The presence of ingredients does not solve the harder mystery of organization, memory, and persistence over time. So the next clue is not where ingredients came from, but how they stopped drifting apart. Volcanic gases, dark clouds, harsh rain, and heat gave early Earth a restless chemical atmosphere above. Imagine standing under that ancient rain watching minerals wash across black rock where nothing yet lived. Lightning was violent, but its energy could rearrange simple gases into more complex chemical pieces below. In modern laboratories, scientists recreate small parts of those conditions, never the full ancient planet itself. Energy matters because it can break ordinary molecules apart and let them recombine in unusual ways. These experiments show plausible chemistry, not a recording of exactly what happened 4 billion years ago. Making organic compounds is only the beginning. Turning them into life remains a much deeper puzzle. A garage floor covered with tools is not a working engine until something connects the parts. Rainwater and runoff could compounds from volcanic slopes into pools, cracks, and temporary basins below. But spreading chemicals through water creates another problem. Useful ingredients become too diluted to interact meaningfully. A dark meteorite fragment can preserve from chemistry older than any rock on Earth today. Researchers study these stones like chemical archives, asking what raw materials the young planet received from space. Carbon compounds inside meteorites are not signs of living things. They are ingredients, not biology itself. That distinction matters because organic chemistry can exist in space, deserts, and laboratories without life anywhere. At impact sites, space-borne material could mix with Earth's own rocks, water, heat, and minerals below. Dust, glassy fragments, and dissolved minerals may have released useful compounds into nearby pools and cracks. Once released, those compounds would scatter unless surfaces, cycles, or boundaries helped gather them again locally. Could mineral-rich landscapes have acted like primitive sorting tables for early Earth's restless chemistry and debris? The same unstable world delivered ingredients, shattered rocks, boiled water, and destroyed fragile molecules repeatedly too. The next clue lies in places that repeatedly filled with water, dried out, and filled again. In shallow pools, chemistry did not stay constant. Sunlight, steam, and minerals kept changing the mixture. As water evaporated, dissolved molecules could crowd into thin films along mud, salt, and rock surfaces. Crowding matters because molecules that rarely meet in open water can react when forced together more often. It is like reducing broth on a stove. Concentrating flavor without pretending soup becomes alive somehow. Drying could help organize chemistry, but it could also damage delicate compounds before they mattered chemically. The important part was the cycle. Water vanished, chemistry concentrated, then water returned to remix it. When rain or tides came back, concentrated compounds could loosen, move, and meet new partners again. Modern experiments test these wet-dry cycles, using controlled settings to explore possible prebiotic pathways today carefully. The results are clues, not verdicts. No experiment perfectly reconstructs the first living moment on Earth. Even organized chemistry needed somewhere to persist instead of dissolving back into the restless landscape again. Layered minerals offered structured surfaces where drifting molecules might collect, align, and remain nearby longer together. A surface can hold molecules close enough for reactions that open water would rarely allow naturally. Order did not require intention. Sometimes the shape of rock itself can guide chemical behavior quietly. Across early landscapes, countless tiny cracks and grains may have acted like natural chemical workshops everywhere. Flowing water could bring fresh material in while carrying away byproducts that blocked further reactions nearby. In labs today, mineral samples help test how surfaces influence prebiotic chemistry under controlled conditions carefully. Inside tiny pores, chemistry could become trapped, protected, and concentrated in spaces smaller than dust grains. But organization still needed power, because useful patterns fade unless energy keeps chemical reactions moving forward. The story now shifts from shallow pools and mineral films toward deeper, darker energy sources below. Beneath the ancient ocean, another kind of engine may have been waiting in the dark below. The trail of raw ingredients now sinks toward darker water, where geology may provide the next missing force. Organic molecules can drift for ages, but without energy and contact, they remain chemistry waiting for direction. Below the ancient ocean, the mystery expands from tiny particles to a vast engine of rock and heat. Could a world of vents and minerals have pushed scattered molecules towards something more organized over time? Along rough mineral walls, tiny pores may have offered early chemistry a place to gather and react. Hydrothermal vents are not a proven birthplace, but they remain one of science's strongest possible settings. Here, sunlight disappears completely, leaving pressure, minerals and hot fluid to shape the seafloor world below. When superheated fluid meets cold seawater, invisible differences appear, creating sharp chemical and thermal contrast around rock. A chemical gradient is simply a difference, but differences can push reactions in useful directions through time. Inside mineral pores, molecules may have stayed close long enough for repeated reactions to matter more. A tiny pocket in rock could trap chemistry that open seawater would quickly scatter and dilute. At this scale, one pore may matter like a room inside a skyscraper-sized natural reactor below. Mineral surfaces can hold molecules nearby, increasing the chances that certain reactions happen again and again. But the same vent energy that drives chemistry can also destroy fragile molecular arrangements very quickly. The safest chemistry may not happen in the hottest plume, but along its protected mineral edges. A vent field is not one machine, but a network of pores, gradients, and flowing water. Constant energy flow can keep chemistry from settling down, allowing temporary patterns to appear and persist. Modern instruments can visit vents today, but they capture only fragments of what ancient Earth allowed. A narrow fissure becomes a moving border, where two kinds of water meet and react continuously. Gradients may power reactions, but the harder question is how any useful pattern could last there. This alien-looking world begins with ordinary seawater slipping into rock, heating, reacting, and rising again through darkness. Heat can speed up chemistry, yet too much heat can tear delicate molecules apart rapidly nearby. As vent fluids cool, minerals fall out of solution, building walls, layers, and hidden pores below. Those surfaces could concentrate ingredients, separate them, and expose them to changing chemical conditions again nearby. The environment itself may have shaped the chemistry as much as the molecules drifting through it. Some textures resemble connected rooms, where reactions could be separated, repeated, and compared by nature itself. If fluid circulated through those rooms again and again, random chemistry might become less random over time. Across a chimney wall, warm interior fluid and cold ocean water could create persistent gradients for reactions. Submersibles can sample modern vents, but ancient prebiotic chemistry must be reconstructed from scattered geological clues. The vent hypothesis is powerful, yet it still leaves the birthplace of life unresolved today by evidence. So the investigation keeps vents in view, while turning toward other possible chemical landscapes on Earth. Life's beginning may not belong to one setting, but to several environments, contributing different advantages. On ancient shorelines, drying pools could concentrate molecules in ways deep water could not easily do. When water evaporates, molecules crawl together, making some bonds more likely than they were before drying. Small pools along early coasts may have repeated this cycle through rain, heat, and evaporation many times. The evidence keeps several doors open: pools, vent gradients, mineral surfaces, or combinations of them. Early Earth may have held countless chemical laboratories, from impact craters to coasts and seafloor vents. Movement matters because useful chemistry needs fresh ingredients, waste removal, and repeated encounters over time together. A steady flow through one tiny pore could prevent reactions from stalling into chemical silence completely. But energy and motion are still not enough unless patterns can survive from moment to moment. The next mystery is how chaotic reactions could begin favoring chemistry that repeats itself over time. A life-like network needs products that help keep the reaction system going, even briefly, before fading. These glowing loops show a hypothesis, not a replay of life, actually beginning on ancient Earth. In laboratories, scientists recreate vent pressure, minerals and fluids to test which reactions are plausible today. Experiments can reveal possible pathways, but they cannot rewind Earth to the exact first event itself. Any early system faced disruption from heat, dilution, shifting chemistry, and constant environmental instability around it. Sheltered microenvironments may have given fragile chemistry more time to persist, change, and interact repeatedly there. Across a young planet, billions of protected pockets could multiply the chances for useful chemistry enormously. Now the story moves from energy sources toward boundaries that might hold chemistry together in one place. Energy may start the process, but boundaries may decide when chemistry gains an inside and outside. The curved edge of a droplet becomes a mineral pore, linking soft chemistry to ancient rock in darkness. Along the vent wall, warm chemical fluid meets cold ocean water, creating differences that may matter deeply. Inside tiny pores, drifting molecules slow down, gather together, and encounter mineral surfaces more often than outside. A chemical gradient is simply a difference across space, and differences can push reactions forward over time. Modern labs can recreate parts of this world, though never the full ancient ocean itself under pressure. Outside the microscope, the same question stretches across a chimney, walls larger than any single controlled experiment. Flow alone can concentrate chemistry, turning rough mineral geometry into a possible organizing force before biology. A pore network may have acted like a maze, holding molecules longer than open water ever could. Heat flow and concentration may have changed repeatedly, testing chemistry without direction or biological control through time. Energy could move ingredients, but the next mystery is how any of them became organized together. Before cells existed, boundaries could appear as rock walls, films, droplets, or mineral pockets in water. A simple edge can create an inside and outside, even before anything is truly alive at all. Experiments test these boundaries under simplified conditions, revealing what physics can do before biology begins in nature. No single setting owns the mystery. Vents, pool minerals, and cycles each offer different clues worth testing. Energy fills the scene with motion, but organization may begin where chemistry finds a lasting edge. This round compartment is not life, yet it changes what can happen inside its fragile boundary. Some molecules naturally line up at surfaces, forming fragile edges without any biological instruction or design. When many droplets appear together, self-organization begins to look strangely purposeful, though it is not alive. The boundary, the mineral surface, and the surrounding water may have shaped one another, together slowly. Outside and inside are no longer the same, and that difference changes the chemical story unfolding here. When compartments touch, their contents can mix, separate, or shift in ways open water cannot easily. Most early compartments would fail quickly, torn apart by heat, motion, dilution, or unstable chemistry. Rising and falling water may have repeatedly concentrated ingredients, then release them into new mixtures again. When ingredients become crowded, rare reactions can become more likely than they were in open water. A rounded shape can resemble a cell, But shape alone is not evidence of life itself. Temporary internal conditions may still matter, because brief differences can change which reactions happen. Next, inside. A boundary is not only a wall. It can also filter what enters and leaves behind. Holding ingredients together gives chemistry a place to continue, instead of disappearing into the surrounding ocean. For chemistry to move closer to life, useful patterns must somehow last or return again later. Droplets and mineral surfaces may have worked together, each helping stabilize what the other could not. A shallow groove can guide when a landscape directs rainwater, without any living navigator involved. Repeated flow through the same small space can make organization appear from ordinary physical motion alone. Stability becomes a serious problem, because a useful structure means little if it vanishes immediately afterward. Persistence may depend as much on the environment as on the molecules held inside each compartment. Early chemistry needed time, not just ingredients, because countless failures may have come before progress appeared. The edge of a compartment can shape reactions, making the boundary more than empty packaging alone. Too much isolation would trap chemistry, while too much openness would wash everything away too quickly. Early environments likely shifted constantly, with heat, minerals, water, and drying repeatedly, changing the rules around the chemistry. Warm and cool cycles may have shaped reactions by repeatedly loosening and tightening chemical arrangements over time. The path toward life may have depended less on one moment than on repeating cycles instead. Across the mineral surface, repeated droplets create pattern spaces where chemistry can linger longer than expected. When a compartment lasts, its internal pattern becomes visible enough for the next question to emerge. Persistence is not inheritance, but it points toward the harder problem of chemical memory beyond simple stability. The boundary can shape the contents, while the contents may slowly alter the boundary itself over time. Many pathways likely collapsed, leaving unstable chemistry behind before any lasting system could emerge over time. Here, survival means physical persistence, not biological fitness, and that distinction matters quietly throughout this sequence. The camera narrows into the channel, carrying us from compartments toward internal chemical patterns waiting ahead. The next mystery is whether patterns can persist, influence reactions, or begin to repeat over time. Chemistry is nearing the idea of memory, but it has not crossed into biology in any true sense. Compartments created an inside. Now the story asks what could make a useful pattern last longer. Once chemistry has an inside and outside, reactions can become concentrated instead of vanishing into open water. A single protocell-like droplet drifts through mineral-rich water, carrying suspended molecules through the restless ancient pool. Modern lab glass lets researchers test small pieces of conditions that early Earth may have provided naturally. This boundary is not alive, yet it changes the odds for every reaction trapped within it. At this tiny scale, heat, drying, and minerals turn the whole landscape into a chemical engine. When water molecules crowd together, making rare reactions more likely to meet the right partners. A compartment is like a temporary room, giving chemistry fewer exits before the next disturbance arrives. But holding molecules in one place means little unless useful patterns can somehow survive the next cycle. The camera leaves one droplet and follows repeated curves spreading across the same wet mineral surface. What happens when a fragile compartment swells beyond what its own boundary can safely hold together? The membrane stretches and trembles, showing how internal chemistry can push against a delicate outer edge. Growth, like motion, can happen for simple physics alone, long before anything should be called an organism. If the boundary leaks too freely, concentrated chemistry dissolves back into the surrounding ancient water again. The best compartment would need a difficult balance, open enough to exchange, closed enough to endure. Tiny mineral grains may steady the edges, giving fragile droplets places to grip instead of tearing apart. Scaled up, this resembles controlled traffic through gates, not a sealed jar locked away from the world. Researchers can recreate fragments of this puzzle, but never the full chaos of early Earth itself. The harder question is whether any fragile compartment could carry a chemical past into its future. The visual trail now moves from protective boundaries toward repeated patterns forming inside them for the first time. Inside the protocell-like space, faint arrangements appear and vanish faster than a human heartbeat in darkness. Shelter matters, but life also needs a way for useful arrangements to last beyond one moment. The mystery shifts from walls to memory, from simply holding chemistry to preserving useful information through time. A repeated molecular shape clings to the mineral surface as the shallow water slowly recedes again. This drying pool becomes the bridge to a deeper question. How can repetition become inheritance later? Could early Earth have found a molecule that carried instructions while also helping reactions occur nearby? In laboratories today, Runner draws attention because it can do more than merely store chemical information. Runner can carry sequence information, and some Runner molecules can help chemical reactions move forward under certain conditions. That does not prove Runner came first, but it explains why the hypothesis remains scientifically powerful. If information entered here, chemistry was no longer only reacting. It was beginning to remember small differences. The same ancient pool now asks a different question: which patterns can survive repeated disturbance over time? A strand, like a mineral surface, where smaller pieces gather around it in moving water. When building blocks meet the same template again and again, copying, like chemistry, becomes easier to imagine. But a plausible pathway is still not proof that this happened on the real early Earth. Experiments test pieces of the puzzle, from nucleotide formation to molecules that influence nearby reactions in solution. The crucial step may not have been perfection, but persistence through heat, water, and constant change. Across countless droplets, rare successful patterns would have been surrounded by far more chemical failures and false starts. One faint pattern remains inside a droplet, while the surrounding pool darkens and cools around it. A lasting molecule is not alive, but it changes what the next chemical cycle can inherit. The scene turns from storing information toward the harder problem of copying it under unstable conditions. How could early chemistry copy anything, while heat, water, and minerals kept rearranging the system again and again? A surge of water scatters loose molecules before the fragile pattern has time to stabilize again. Early copying did not need perfection. Preserving even part of a structure could matter over repeated cycles. In a strange way, mistakes may have opened different futures for the same chemistry over many cycles. Researchers search for plausible steps where simple molecules copy, fold, or influence reactions around them in water. Across enough repeated cycles, even tiny advantages could begin separating persistence from complete disappearance over time. One strand survives inside a droplet, while another fades into scattered pieces nearby in the water. Chemical memory begins when the past slightly changes what can happen in the next repeating cycle. If that memory strengthens, the border between lifeless chemistry and biology becomes harder to draw precisely. The next clue follows compartments where information, boundaries, and energy begin interacting together in one system. Copying like chemistry, could preserve patterns before modern DNA, giving early molecules a fragile kind of continuity. On mineral surfaces, molecules stay close together, allowing reactions to repeat instead of drifting apart in water. Information may not begin as written instructions, but as patterns that keep appearing in the same arrangement. A pattern matters only when it changes what happens next, guiding chemistry toward another possible step. This strand-like molecule is a guide for imagination, not proof that early runner looked exactly this way. If memory forms inside a boundary, could a tiny compartment begin carrying its own chemical past? The view widens from one strand to many possibilities, each competing for stability in a restless environment. Some patterns last longer because minerals, droplets, or sheltered films protect them from being scattered too quickly. Heat, ultraviolet light, and dilution constantly threaten fragile chemistry, destroying many arrangements before they can truly persist. The trail leaves the lab and returns to early Earth, where every shoreline becomes another test. As water evaporates and returns, a shallow pool concentrates molecules, then challenges them to survive change. Repeated drying may help molecules link together, while refilling decides whether those fragile structures can still hold. A thin mineral film can become a temporary reaction zone, even without anything like a modern cell. This pool is not just water on rock. It behaves more like a changing natural reactor. A longer chain means the less it can influence future chemistry, helping one pattern affect another. The threshold is still not life, but chemistry may be learning how to preserve an advantage. Inside a vesicle, reactions remain close enough to influence one another again and again over time. A microscopic boundary floats inside a violent planet, surrounded by waves, heat, impacts, and deep darkness. Most fragile structures probably failed, breaking apart or dissolving before anything stable could continue for long. But failure can act like a filter, leaving behind arrangements that endure just a little longer. Before biology, selection can begin when some chemical systems simply persist better than their immediate neighbors. One droplet leaks quickly, while another holds its ingredients long enough for reactions to continue inside. We follow this tiny compartment as a possibility, not as a confirmed ancestor of every living cell. A protocell-like system matters only if growth, division, and internal patterns can repeat together in sequence. Too much change destroys the system, but too little change prevents any path toward real adaptation. The deeper mystery is coupling boundaries, energy, and information beginning to work as one connected system. If those pieces joined, would we still call it chemistry, or something beginning to live at last? The story expands beyond one droplet, toward environments where countless chemical experiments could unfold at once. Before ecosystems, early landscapes may have held trials, failures, and brief successes too small to see. Now the question changes. What happens when chemistry stops acting alone and begins acting like a system? The first living systems may have nothing like animals, plants, or the familiar cells we imagine. A thin film across stone suggests organization, but it does not let us see early biology directly. Early metabolism may have moved energy through simple reaction networks, long before complex cells existed anywhere. At human scale, life's beginning may thinner than paint spread across ancient stone. The first life was probably not a dramatic creature, but a threshold crossed by connected systems. These tiny networks are possible ancestors in a model, not accurate portraits of the first organisms. Modern explorers study analogs and traces because no camera can return to Earth's first living moment. Layered rocks matter because microbial communities can leave patterns that survive long after the cells vanish. Early systems faced burial, chemical shifts, impacts, and changing oceans before any lasting stability could exist. When the water clears, persistence becomes the clue that something different may have begun here quietly. Repeated growth across a surface can build structures that later become readable in very ancient stone. Bubbles and layers may hint at metabolism, but the exact early pathways remain difficult to reconstruct. Small chemical systems eventually connect to coastlines, oceans, and the larger cycles of a changing planet. A living system is not just material. It manages energy, boundaries, and information together through time. The oldest clues are indirect, forcing scientists to read possible biology through chemistry and rock patterns. A rock sample becomes a time capsule, preserving hints from microbial worlds that disappeared long ago. But how much of the pattern is life, and how much is geology imitating life in stone? That uncertainty matters, because false confidence can turn a promising clue into a story science cannot support. The investigation returns to the sea, where early living networks may have started spreading quietly through water. The next question is how tiny systems became communities capable of changing entire environments around them. The earliest living systems may have looked less like creatures and more like thin communities clinging to wet stone. Across the shallow floor, layered films trap minerals, sunlight, and chemistry in a fragile, repeating pattern. No eyes watch this world, yet every surface is slowly being reshaped by microscopic activity and water. A single mat is not a city, but it begins to act like a connected environment. Water carries dissolved minerals across the layers, feeding reactions too small for any human eye to follow. At this scale, survival is not dramatic. It is chemistry persisting slightly better than before, repeatedly. The boundary between living and non-living becomes harder to see when the system is this simple. What matters is not one heroic organism, but a pattern that can keep renewing itself through time. Layer by layer, the microbial mat turns sunlight, minerals, and water into a more organized world. The first ecosystems may have been thin enough to miss, yet powerful enough to leave traces. Some ancient rocks preserve structures that resemble layered microbial worlds, though interpretation still requires scientific caution. A stromatolite-like mound rises slowly, built not by bones, but by sticky films and trapped grains. Each new layer records a quiet struggle between growth, sediment, light, and moving water over time. For a viewer standing nearby, this would look like stone, not life reshaping an ancient coastline. The clue is not a fossilized body, but a repeated structure that living mats can build. Scientists compare ancient rocks with modern microbial mats, searching for patterns too organized to be accidental. This evidence does not show the first life directly, but it shows life was already changing Earth. The mystery shifts from origin to consequence, from how life began to what life started doing. Even without animals or plants, the planet was becoming biologically active in shallow ancient seas already. A quiet shoreline can hide a revolution when microbes begin altering chemistry faster than rock alone. Sunlight touches the surface, and some microbes learn to use it in a more powerful way. Photosynthesis did not begin as a green forest, but as microscopic chemistry inside ancient water systems. Tiny bubbles rise from a mat, small signs of a planet beginning to change its air. Oxygen seems harmless to us, but for many ancient microbes, it may have been a threat. The same gas humans today could disrupt chemical systems that evolved in an oxygen-poor world. Iron, rich water reacts with oxygen, leaving rusty clues that later become bands inside ancient rock layers. The ocean itself becomes a record, storing chemical evidence of a long planetary transformation in stone. This was not a sudden breath, but a slow planetary shift unfolding across immense geological time. Every bubble hints at an atmosphere that was not yet ready for complex animals or forests. Before Earth looked blue and green from space, microbes were quietly rewriting its chemistry from below. The scale is difficult to imagine because the actors were microscopic and the effects were planetary. A mat only inches thick could participate in changes that eventually shaped the entire biosphere. Above it, the first living systems did not conquer Earth with speed, but with persistence across many generations. Their success came from network surfaces and repeated reactions, not from brains, muscles, or movement alone. Life was becoming a force of geology, leaving subtle marks that stone could preserve over time. Yet every preserved clue also asks how much earlier the story began beyond the surviving record. Ancient rocks are not perfect archives. Heat, pressure, and time can erase fragile biological evidence completely. That is why scientists handle early life claims carefully, weighing structure, chemistry, and geological context together patiently. A beautiful pattern in stone is not enough. It must survive skeptical testing from multiple directions. The oldest life story is written in fragments, and every fragment changes how we read Earth. By the time microbial communities spread widely, Earth was no longer just rock, water, and air. The planet had become interactive, with biology feeding geology and geology shaping biology in return continuously. This feedback may be one reason life did not remain a fragile chemical accident for long. Once living systems could modify their environment, they also created new opportunities for future life forms. The first ecosystems were small, but they opened a path toward a more complex living planet. From thin mats and mineral layers, the story eventually leads toward oxygen, cells, and evolution itself. But the first chapters remain partly hidden, buried beneath billions of years of planetary change below. What we can see is enough to know life became active early in Earth's history. What we cannot see is the exact moment chemistry first crossed the threshold into life itself. That unanswered moment carries us forward from ancient microbial worlds toward the planet they transformed later. Along this shallow ancient coast, life is not a creature yet, but a dark texture under moving water. The mat spreads in layers across the shallows, turning sunlight, minerals, and water into a living surface. Tiny bubbles gather along the ridge, making visible metabolism visible for just a moment before drifting away. Multiply that small surface across miles of coastline, and a quiet process begins, reaching planetary scale. Layer by layer, sediment and microbial growth build a record no eye could have watched forming. Some microbes learn to use sunlight as power, changing water and carbon into new chemical possibilities. The organisms reshaping Earth were not dramatic giants, but fragile films clinging to mud, rock, and water. Each bubble trail asks a harder question: what hidden chemistry was happening inside these ancient surfaces? A shoreline smaller than a neighborhood could connect local microbial work to the future air above Earth. But oxygen entering an older chemical world would not feel like a gift to everything alive. Beyond the mat's iron-rich water waits like a silent chemical trap spread through the ancient sea. When oxygen met dissolved iron, it could lock the reaction into minerals that survived deep time. Before oxygen filled the atmosphere, Much of it disappeared into oceans, rocks, and other chemical sinks. For life adapted to oxygen-free worlds, this new gas could be stress, danger, and disruption in every surrounding habitat. Anaerobic habitats faced a changing chemistry, where familiar waters slowly became harder places to survive over generations. Local reactions repeated across ancient seas, turning scattered microbial activity into a wider planetary transition over immense time. Imagine a national park cliff as stacked pages, each layer holding a different chapter of Earth. In banded rocks and mineral crusts, the planet preserved clues from transitions too slow to witness. Microbial surfaces did not just live on Earth. Over time, they helped rewrite Earth's chemistry. If oceans were absorbing oxygen first, when did the atmosphere itself begin to change above the planet? From above, scattered microbial shorelines beam a thin living edge, wrapped around a young and changing planet. Oxygen had to fill Earth's chemical sinks before it could remain freely in the air. A planet becoming breathable for us was also becoming hostile for many earlier forms of life. Mineral crusts, layered deposits, and altar rocks become witnesses, even when ancient cells are long gone. Through simple metabolism, microbes connected sunlight, water, carbon and oxygen in a cycle with planetary consequences. Each rock layer is like a page stacked under another, turning deep time into something visible. A film of minerals thinner than paint can represent centuries, storms, and countless microscopic chemical events. This transformation was not a sudden switch, but a long, uneven shift across oceans and air. So what does it really mean for a planet to become alive, not just inhabited by organisms? If life can alter an atmosphere, that change becomes a clue for worlds beyond Earth as well. From space, the microbial story becomes larger than any shoreline, reef, or single fossil, like traced below. Ancient coasts, shallow seas, and mineral beds together form a scattered evidence network across the planet. A biosignature does not need a skeleton. Sometimes it is chemistry left behind by vanished life. The oldest clues are often fragmentary, indirect, and debated, which makes careful interpretation scientifically essential today. Modern scientists read ancient rocks with instruments, models, and caution, searching for patterns without forcing certainty. Under the microscope, tiny textures can hint at biology, but they rarely speak alone with certainty. Living surfaces constantly exchanged material with water and sediment, slowly turning activity into preserved structure over time. A bubble smaller than a raindrop can belong to a process that eventually changed the sky. By now, microbes, rocks, oceans, and air have become one connected story of planetary transformation unfolding. And if traces of life can survive here, where else might hidden evidence be waiting now? The ancient shoreline remains quiet, but it marks a turning point in Earth's long becoming. Seen from orbit, Earth's transformation is no longer local. Biology has entered the planet's appearance. A living world can leave fingerprints in air, water, and stone, even after organisms vanish completely. Earth's oldest mystery has become a search method for places we have never walked or sampled. The same question now turns toward worlds that look lifeless, but may still hold the ingredients. If chemistry crossed the line once on Earth, could it cross that line somewhere else in darkness? Modern exploration searches not for certainty first, but for water, energy, chemistry, and the right context. Across other worlds, rock and water may meet. Energy in forms Earth can help us recognize. The haunting question is whether similar ingredients can organize themselves again beyond our planet and our history. From early Earth, the story now widens toward Mars, ocean moons and distant worlds still unread. Early Earth does not give us a final answer, but it gives every search for life a beginning. On Mars, dry riverbeds suggest water once moved across a world that may have shared Earth's ingredients. A rover's wheel marks the dust, while instruments search for chemistry that ancient rocks might still remember. Beyond Mars, Europa's cracked ice, Haitian, where sunlight may never reach the water below. Enceladus throws bright glimmers into space, carrying clues from a buried ocean humans have never entered. Around distant stars, small planets dim their suns, hinting at worlds we can barely describe yet. Astronomers read those tiny shadows like fingerprints, searching for atmospheres that might hold subtle chemical imbalance. Back on Earth, old rocks become our guidebook for worlds we cannot touch. The same question follows every probe and telescope. What conditions let chemistry begin acting almost alive? Water may be common, energy may be common, but organization is the part we still struggle to explain. Early Earth reminds us that life did not need forests, animals, or oxygen to begin changing a planet. It may have started in darkness, heat, minerals and boundaries too small for any eye to see. Every hypothesis leaves gaps, but together they reveal a path from raw chemistry toward living behavior. The mystery is not a missing creature, but a threshold where matter began preserving useful patterns. That threshold may have been crossed slowly through countless failures that left no fossil trace behind. By the time rocks clearly record life, the first experiments may already have been ancient history. Stromatolites and microbial traces are not the opening scene, but surviving echoes from a deeper beginning. Later, tiny microbes would help reshape oceans, skies, and every future breath taken by complex life. The transformation took immense time, yet the earliest clues suggest life appeared surprisingly early on Earth. That timing keeps scientists looking outward, because a quick beginning here may mean other beginnings elsewhere. Still, Earth remains the planet where chemistry is known to have become biology so far. That single example makes question both fragile and enormous, like holding one match in darkness. Mars, Europa, Enceladus, and distant exoplanets are not answers yet. They are testable scientific possibilities for future missions. Each mission returns to the same ancient ingredients: rock, water, energy, chemistry, time, and a boundary. If those ingredients meet again, the universe may have places where chemistry is still quietly trying. But science cannot replace evidence with hope. So the search moves carefully, clue by careful clue. The first living systems may never be reconstructed completely because their world has been erased by time. What remains is a trail of conditions, patterns, and consequences written across the planet itself today. A lifeless Earth became alive once, and that fact still changes how we see every world. The deepest mystery is not only how life began, but how often the universe gets another chance.